Hello, good morning. My name is Naveen Kumar Viswan, and I'm going to talk about our university project named as Cellular Antioxidant Potential, Anti-Inflammatory and Cytotoxic Activities of the Extracellular Polysaccharide isolated from Lactobacillus graminis strain KNU-AS018. So in this presentation, we will going to show what we have done in this project and what we have achieved and where we have submitted the article. Like we can see here, these members are our the team members, and my name is Naveen Kumar Viswan, and whatever the findings we have got from this project, we have submitted in the journal Biocatalysis and Agriculture Biotechnology. The current status of the article is it is under review. The impact factor of the journal is, I think, more than 4. If we talk about the background information or the introduction about this manuscript, why we needed to do this kind of works. So we know that WHO has reported that almost 74% of the deaths are due to the non-communicable diseases, and they have really severe diseases like respiratory diseases, cardiovascular diseases, diabetes, and cancer. Non-communicable diseases are associated with some free radicals called reactive oxygen species, reactive nitrogen species, and they are generated with the physiological progresses in the body. And when they are generated, they lead to the oxidative stress and generate several diseases such as mellitus, Alzheimer's disease, Parkinson disease, cirrhosis, and even depression also. For that, some antioxidants molecule which can inhibit the ROS and RNS activity within the body and can relieve the cellular oxidative damage. On the basis of their production in the natural system, we can see some of the antioxidant work as endogenous and some of the work as exogenous, and they help to maintain the redox balance within the body naturally produce some antioxidant also, but in the disease condition or in the challenge conditions, some antioxidant supply needed to be given the body for the management of the diseases. We know the excessive ROS production in the body lead to the inflammation, and inflammation is also related to the several diseases and which need to be tackled via the suppression of the reactive oxygen species within the body. And we know some extra when we do the antioxidant supplementation in the body, it can give some adverse effect also to the mammalian system. That's why whenever we check the natural product for the biotherapeutic use, we need to check the toxicity analysis in advance. So we are coming to the our basic topic, which is the polysaccharide. Why we have chosen polysaccharide? Because polysaccharide are some insoluble polymers. Sometimes it can be soluble in the water, sometimes depends on their polarity, it can be soluble in the some organic solvents. They are produced by the microorganisms and then released in the extracellular medium. It has been reported that they help in the management of inflammation and oxidative stress related diseases via inhibiting the activity of T cell, which is a very important cell of the immune system. So we have when we are working on the polysaccharide, we decided to take the microorganism such as lactic acid bacteria, because lactic acid bacteria generally considered as non-toxic and are reported to produce several biotherapeutic compounds, and can sometimes they are used as the probiotic product also. So based on this all background information, we have chosen lactobacillus strain for the production of extracellular polysaccharide, and then we have tested that polysaccharide for their antioxidant activity, anti-inflammatory activity, and cytotoxic activity in non-cancerous cells. And we are going to talk about the findings given by this study. In this slide we can see the total methodology used, and the graphical abstract of the uh, total work is also presented here. This slide represents the extraction and fractionation of the polysaccharide from lactobacillus. We can see here on the column chromatography elution profile sees the three kind of fractionation peak in the polysaccharide: EPS1, EPS2 and EPS3, depending on their solubility in the NaCl concentration. Such as EPS1 come with the water solution, but EPS2 and EPS3 come with the gradient NaCl concentration. Then lately we did the purification and biochemical characterization of the fractionated polysaccharide. We can see here this picture A, figure 2A represents the EPS1, B represents EPS2 and C represents EPS3. These all peaks shows a single symmetrical peak, which also shows that the no impurity were or no another polysaccharide fraction was eluted with these fractions. And then we have checked their total yield concentration was 14% for EPS1, 9% for EPS2 and nearly 1% for EPS3. The trace amount of TPC and TFC was found in EPS1 and EPS2, but no TFC were detected in EPS3. However, the nucleic acid were absent in all the fractions, and the trace amount of protein was detected in the EPS3. And the figure 2D also represents the no significant absorbance on 260 to 280 nanometer, which also suggests that the EPS fractions was free from the adulteration of nucleic acid. Then further we did the FTIR analysis of our three polysaccharides, and presence of the transmittance peak, we can analyze the presence of the biomolecules on the surface of the extracellular polysaccharides we have found, and on the basis of these peaks we can identify which functional group present on their surface. For example, nearly 3200 wave number, they are representing the OH stretching of the molecule, and then later 2900 it represents the CH stretching. Similarly, we can identify all the transmittance peak and assume that what functional group present over there. Then in the bioactivity analysis, we check the free radical scavenging activity of our sample against DPPH and ABTS radical, and also compared our findings with standard ascorbic acid. We can see here in the first graph DPPH radical scavenging activity. The EPS1 shows the higher scavenging activity when the concentration was 1 mg per ml, and it was nearly 40%. However, ascorbic acid shows the maximum activity, but that is a synthetic chemical and used as a drug for treating the various disorder. Then figure 4b indicated the ABTS radical scavenging activity. This is the very interesting result. Here we can see 1 mg per ml concentration. The EPS1 shows nearly similar antioxidant activity to the ascorbic acid. However, EPS2 and EPS3 did not show significant activity. In that, EPS3 shows the least one. Since the EPS3 was not higher in the yield and did not show significant antioxidant activity, we went ahead with only two polysaccharide, EPS1 and EPS2, for further analysis. Here in cytotoxic analysis, we chosen two cells, that is NIS3T3 cell and RAW 264.7 cells for the cytotoxic analysis, because both of the cells are non-cancerous in nature. And we can see here the figure 5a represents the cytotoxicity against the NIS3T cell, and we can see EPS1 and EPS2, even 1 mg per ml concentration dose did not cause more significant reduction in the viability. And that also confirmed by the light microscopy images, and we can see here figure 5c represents the same cellular morphology and cell number as compared to the control cells. Similarly, when we check the cytotoxicity in raw cells, we can see the EPS1 and EPS2 treatment somewhere inhibited the proliferation of the raw cells, means they have did some reduction, but that reduction, even 1 mg per ml concentration, was just nearly 35%. That we confirmed by the light microscopy. We can see the EPS1 and EPS2 treatment, the little inflammatory bodies were found as compared to the control cell. Then for cellular antioxidant activity, we know that AAPH is a azo compound, and it generates intracellular free radicals and then in turns the DNA damage and apoptotic cell formation when it is given to the cellular system. That's why we check the cellular antioxidant activity of these two samples against AAPH induced antioxidant AAPH introduced intracellular free radicals in NIS33 cell. We can see here in the graph figure 6a, when AAPH was induced, the viability was reduced to nearly 55%, but when the EPS1 and EPS2 concentration were given, the viability was significantly increased. When the medium dose of the sample were given from 125 to 250 mg per ml, we can see the cell viability was nearly 90%. The higher concentration did not show the highest activity, because maybe when APH and sample polysaccharide sample mixed together, they can also generate a little bit of oxidative stress. That findings were confirmed by two staining images, AOAB staining and DCFHDA staining, and we compared with the control. We can see the APH induction induces some apoptotic body changes and some necrotic cell in the red color we can see, but when EPS1 and EPS2 treatment was given, it was found nearly to the control cell. Similarly, the DCFHDA staining shows that APH induction induced a high amount of ROS status, while EPS1 and EPS2 inhibited the reactive oxygen production in the cell. Further, for anti-inflammatory activities, we have chosen raw cells, because raw cells are the macrophages, and we know the macrophages are the essential components of the host defense mechanism against several diseases, even in the microbial infection. And the macrophage-like cells such as RAW cells are most often used in anti-inflammatory disease, because when the foreign particle is given to the body, the macrophages are the ones which shows the inflammation response. Here also we see the AAPS treatment to the cell reduced the cell viability nearly 45%, but the EPS treatment given in the concentration significantly increasing the viability compared to the AAPS treatment. At 250 mg per ml concentration, EPS1 shows the highest cell viability, that was nearly 75%, which were also confirmed by AOAB staining and DCFHDA staining. We can see the AAPS treatment to the raw cells have shown several inflammatory body in the cell, and necrotic cells also can be seen in red color. But when EPS1 and EPS2 were supplemented, the necrotic body formation was reduced, and ROS status was also reduced in the cellular system. Then further, as we discussed in the beginning, in the introduction system, that toxicity is also a major concern when we testing natural compounds. That's why we tested the blood biocompatibility of these samples. We can say the cytotoxicity on the red blood cells were also tested, and the results were very interesting, because from 15.6 to 1000 mg per ml concentration of both the sample did not show any cytotoxic impact on the plasma membrane of red blood cells. We can see here that Triton X100 was taken as a positive control, that shows the 100% of the red blood cells damage, but the sample causing cellular damage was equally similar to the PBS, means all the cells only shows the nearly 5% of hemolysis, which is quite acceptable, and which shows that both the samples were not toxic to the erythrocytes. Then further, we have compared our results with the previously reported literature also, which have used the lactobacillus as the source of EPS production. We can see here the our strain used for DPPH and ABTS radical scavenging, then AAPH is raised the NIS33 viability, and then AAPH induced raw cells cytotoxicity, and then anti-inflammatory and antioxidant activity was also tested, which have not been reported in some previous literature. We have taken the consideration last 5-year paper. So this is the finding of our project, and we have written the manuscript and we have submitted in the journal. Now it is in the review process. We hope that we can publish this paper. Findings of our project we have taken from the university. Thank you very much for your attention. Thank you.